All right, people. Happy something day. Tuesday. Missing a bunch of people here. What's going on? Bendek, you in here? Claire? Julia? Julia Zhang, are you in here? Miss Angie. All right, so I'm missing Julia, Olivia, Claire, and Sophia. If you're in here, say something. Dude, man, that's not Miley Cyrus, that's Sia. You know nothing about music, obviously. All right, okay, here's my goal. I'll tell you what my goals are, my hopes and dreams. Need some stuff from you guys too, so let me organize my thoughts here. My goal is this. Today, I knock off chapter 9. We finish electron transport chain, rest of chapter 9, regulation of respiration, anaerobics, stuff like that. A few more things to finish up. It'll probably take us the hour. After that, so that's today, that's Tuesday. So then Friday, we're going to cover the first half of photosynthesis. Tuesday next week, I will finish chapter 10, photosynthesis. Friday next week, which is not this week, but next week. So Friday the 18th, we're going to have the next test. So a week and a half from today, we're going to have the next test. It's going to be on energetics. It's just 2 chapters. It's just respiration and photosynthesis. There's only one minor problem with my plan, in that I don't have a plan yet for how we're going to do the labs we're supposed to be doing. Usually I do 2 labs in this unit, a respiration lab and a photosynthesis lab, which are both awesome labs. And I haven't found an online alternative yet. The Pivot thing doesn't have any really good labs that I love. The first 2 I thought were really good, and now these other ones. So I'm going to figure this out, and I might make you do something at home. I don't know. We'll see. But my goal is to have this test on that Friday, because then we can start genetics before winter break and wrap up genetics in January and be exactly where I want to be with our class by the end of the semester, which would be like amazing if I could pull that off. So I'm going to need you guys to, to be diligent and to be motivated for us to get that done. So we're going to go, kind of fast with stuff. And I need people to be, on the ball, and I need people to be present. And I don't just mean sitting, logged into Zoom, but end of the semester is like middle of January, like January, I don't know, 22nd or something. I don't know, whatever, like 3rd-ish week of January. So we'll have like 2-3 weeks, 2-3 weeks for genetics, which usually I can pull that off. Oh, there's Julia. Okay, cool. And there's Claire. All right. So anyway, I need you to be like paying attention in class, even if it's hard and even if it's complicated and frustrating. I need you to, we're going to have, before winter break, I'm going to give you free response practice questions. You're going to get like 25 free response practice questions that are going to prepare you for the midterm. End of January, we're going to have the midterm. It'll be, we'll have a genetics multiple choice test, and then free response for like the whole semester stuff, covering everything. Kind of like honors biology end of the year. You know how I gave you like a whole bunch of like written questions, except this is like honors biology on crack, you get the idea. Anyway, I don't know if I'm allowed to say that, so just strike that from the record. So anyway, don't smoke crack, kids. Drugs are bad. Wait, when is this test again? Okay, I'm going to wait. I'm going to tell you when the test is again. It is a week from this Friday. So whatever that date is that I said, I don't remember now, 18th or something. Here, it's 18th. Okay. And we got somebody else coming in. Sophia. Everybody's showing up. Claire, come on, get in here. I got a new Zoom link. Should work. All right, next item before I start diving headfirst into oxidative phosphorylation. Okay, this is my fault. I neglected to make this clear with you guys, and this is totally my fault. I went into the old Zoom. Okay, sorry about that. Go in the new one, yes. Way back when we did the signal transduction Jamboard, and I allowed you guys to work with groups. Many of you turned that in. They look very nice and everything. The only thing is, like, a lot of them don't have names on the front page. I need everybody's name that was in your group on the front page. So if you could go back into those, here, let's look at them real quickly. If you guys could go back into those, and oh my God, Sia, I just love her. How can you not like Sia? I mean, something's wrong with you if you don't like her. She's the best. Such a good songwriter. Anyway, let's look at this thing. It was called the Signal Transduction Jamboard. So we got, so here's what I'm looking at. I got all these here, I'm just clicking on Michelle Certain, not to try to pick on her or whatever, but see, I don't see the names are here. So Amira B, Michelle Certain, Nevada K. Okay, so the names are in the title. So that's helpful already. So that's good. I just need everybody to make sure they have the names. Looks like Max Egan's has all the names in the front. Allison Hampel. I'm just clicking I just need all those names right in the front. And then the other thing I'd like you to do, and I'm kind of apologizing ahead of time for this. If you, or just one of you in the group, could go back, like the owner of the Jamboard, if you could go back into it And I didn't tell you this the other time, but I should have told you. Vance, I don't know what that means. Is that bad? That's bad, huh? Anyway, if you could save it as a PDF, it would make it way easier for me to look at these. I have not looked at them yet. I've not graded them yet. I just glanced at them. And I'm a little bit behind. I'm sorry. I'm way behind on a lot of stuff right now. Kind of freaking out. But anyway, you probably feel the same way. You're probably in the same boat as I am. But if you could go back in that thing at some point, like in the next couple of days, open it up. And if you could save it as a PDF, I can show you how to do that here. Let's just do here. Let's open up Amira's. I'm going to open it with Jamboard. So the way you do that is you just go to the three dots here, right here. Cannot display jam. I don't know why it's not working, but I go to the three. Oh crap! Why is it not working? Well, anyway, you go to the three dots thing in the Jamboard and say save as PDF, and that's what I'd like you to submit then. If you don't mind doing that, if that's a big hassle, then just don't do it. Just write me a note and say it's too much of a hassle, Volk. You're being unreasonable, and I hate you. And then I'll just look at your Jamboard. It's fine. You can just whatever. So I don't want to stress you out too much more. Okay, speaking of stress, no, this is the opposite of stress because this allows you to look forward to photosynthesis, which everybody loves. I have posted photosynthesis stuff already, so you can look at that already if you'd like to, even though we probably won't get to that today. So that's over here. I put chapter 10 reading guide. We can make that one optional if you'd like if it's too much, to have to do them both. So just do the chapter 9 one. But if you want to do both, by all means, if that helps you, by all means do it. That's posted. Useful stuff. I got like notes here. I got images. I got PowerPoint, all kinds of stuff here. Flow chart, energy flow chart. That's kind of funny. But anyway, so I posted that today for photosynthesis, so that's there for you. All right, what I'd like you to do now is make sure you have pencil and paper ready. Your notebook should be ready to go, and we're gonna continue with respiration. We finished up with the Krebs cycle, which I asked you to memorize. So everybody's working on Pukey, a clown, all of that stuff. So please make sure you continue to work on that and practice that. I wasn't planning on going over it again. If there's a need, let me know. If you need me to go over it again, let me know. But otherwise, I was going to move ahead and take a look at what I call the electron transport chain, and some people call that oxidative phosphorylation. It's the same thing, basically. Oxidative phosphorylation is a fancy name for the electron transport chain and the ATP synthase action. So, all right. We are cooking with gas. What gas, you might ask? Well, we're cooking with oxygen, of course. Okay, so here's our Krebs cycle. I know that looks messy and horrible, but I got a clean one for you available on Google Drive, so you can just use that. And also, you should have it copied already. You should have a nice clean one copied in your notebook. So basically, the big punchline of the Krebs cycle is, first of all, the College Board will not ask you to have the Krebs cycle memorized. So for the test in May, you don't have to have it memorized, just for me. And the big punchline of the Krebs cycle is that it takes pyruvate, which is like half a glucose, breaks it down, chops it up into carbon dioxides. And it takes the energy from the pyruvate molecule and makes NADH. That's the big picture, NADH. Yes, it also makes FADH2, but only a little bit. It makes for each pyruvate 4 NADHs and only 1 FADH2. Okay, so I might as well, since I have this open, I might as well draw on this. Okay, so we make, 1, 2, 3, 4 NADHs for each pyruvate, which means 8 per glucose, which is pretty awesome. We only make 1 FADH2. Plus FADH2 is not as exciting because it only gives us 2 ATPs. It's only a coupon for 2 ATPs, whereas NADH is a coupon for 3. So that's already better. And it makes us 1 piddly ATP per pyruvate, or 2 per glucose. So big picture about Krebs cycle or citric acid cycle, some people call it, is NADH, FADH2, a little bit of ATP, and CO2. Now, CO2 is a waste product. It's not like a desirable product that we want. It's a waste product, so we breathe it out. Our bloodstream takes the CO2 from the cells, brings it to the lungs where it gets exchanged into the air in the lungs, and then we breathe it out. And that's the reason we breathe out CO2. And the reason we breathe in oxygen is something we're going to talk about today. But if anybody asks you like, hey, where's the CO2 produced? That's in the Krebs cycle, in the citric acid cycle. All the CO2 is produced there, and it's when half of that glucose, that pyruvate, gets chopped up, and the CO2 comes out as a waste product. All right, the NADH, the FADH2, then goes to the inner membrane of the mitochondrion, and it does a job there. So let me draw you a little picture. And we'll take a look at what exactly is going on in the next steps now. So let's check it out. By the way, I really haven't heard your voices today. So I don't know if you guys are even like, are you guys hearing me?
Yeah.
Okay,
Yeah.
you guys are real sedate today, and half the people have their camera off. Can I just have them on just at first? You can shut them off later again, just to make sure that everybody's there. Unfortunately, we're having trouble with Claire coming in again, so that issue, I guess, was not fixed by the new Zoom link. Still trying to get her in. Claire, are you in? I don't know. I don't see her. Nope, she's still waiting. Sim and Sanjana, if you could turn your camera on for just a little while, that'd be great. All right, so if we take a look at a cell, here we have the cytoplasm. Here we have the mitochondrion. All right, so glycolysis takes place here. And obviously that makes pyruvate. And that then enters the cell, and that feeds into the Krebs cycle. Now, technically, that first step from pyruvate to acetyl-CoA, people don't really consider that part of the cycle of the Krebs cycle. That's just like the thing that gets it started. So technically, like Krebs cycle proper is really acetyl-CoA feeding in there with oxaloacetate. The pyruvate is like a previous step, technically. I just consider it all in one just to simplify it a little bit. But if you want to be like real, nitpicky about it, some might argue with that. So here we have our Krebs cycle. And then, like I said, the Krebs cycle produces NADH. Obviously it produces CO2 as a waste that leaves. And it produces FADH2. So NADH and FADH2 then go to the inner membrane of the mitochondrion, and that's where that thing that I keep calling the electron transport chain takes place. Those things now take that energy from the pyruvate, which came from the glucose, and they bring that to the inner membrane where it can then get transformed into a different kind of energy. They're actually going to use it to pump hydrogen ions into the membrane. I'm going to show you exactly how they do that in just a second. So a whole bunch of hydrogen ions are going to get pumped into this space right here. Tons of hydrogen ions build up, which is actually going to decrease the pH in there because that's like making it acidic. Hydrogen ions make a solution acidic. So the pH drops in here. The concentration of hydrogen ions is real great. And then the hydrogen ions diffuse back into the matrix of the mitochondrion, and they go through this thing called ATP synthase, which starts spinning and making ATP. And that's powered by that gradient of hydrogen ions. That's the reason we need the NADH and the FADH2 is to produce that hydrogen ion gradient. And scientists a lot of times call hydrogen ions protons. So if you hear me say protons or hydrogen ions or H+, that's all the same thing because a hydrogen ion is a proton. Okay, if you don't believe me, just check out the nucleus of hydrogen and you'll be convinced. All right. Okay, cool. Let's now take a look at details. So here is my homemade diagram that I made about 20 years ago in Microsoft Paint, and it's all like chunky and funny looking, but I thought it turned out all right. It's not so bad. What it does is it takes like artists' diagrams that make diagrams for websites or textbooks and simplifies them. And it simplifies the diagram so that you can draw it. So it makes it doable. So it's not 3D, it's not too fancy, it's nice and simple, and it's certainly doable by a high school student. So I'm gonna walk you through it, and then I'm gonna draw it myself. And I'd like you to draw it. I'd like you to draw it out in your notebook to get used to the process. All right, so we'll go through this part real fast, and then I'll go through my drawing so that you can follow along with your drawing. That's going to happen here. Hold on. Let me just make sure I have it in my next slide. Do I have it? Oh, yes, I do. Okay. So I set it up in PowerPoint with their shapes. And I think it looks a little nicer. But so in the inner membrane of that mitochondrion, so where are we? We are right here in that inner membrane. This is where we are. Okay, we've taken a chunk of that membrane. That means this is the space here, the intermembrane space. And then this is the matrix right here. And the Krebs cycle is happening, you know, right here. The Krebs cycle is feeding FADH2 and NADH to this electron transport chain and powering it. NADH comes in right here at complex 1. That's called NADH dehydrogenase. And it gets dehydrogenated, literally. It gets dehydrogenated. A hydrogen gets pulled away from it. So a hydrogen ion comes off. Okay, we could draw that in here. It's missing from my diagram. And 2 electrons from this molecule, 2 electrons get channeled or funneled, shot into this complex, this NADH dehydrogenase complex, and they get fed into another molecule called ubiquinone. So ubiquinone is right here. That's the U, and that's where those electrons go. Now, in the process, those electrons act like a power source, kind of like for a motor or a pump. Like we would plug in a water pump to pump water out of our basement or whatever, like a sump pump. Well, this is like a different kind of pump. This doesn't pump water, but it pumps hydrogen ions. So it takes a hydrogen ion from here and pushes it into the space up here. All right, and that's powered by those electrons from NADH, because those are high energy. They have a lot of energy in them. And we know electricity can power machines and motors and things like that. Well, that's what's happening here. This electricity is powering this little protein machine. It's actually kind of a big cluster of proteins. It's a complex of a bunch of proteins, which we know what they are. We just simplified for our purposes because AP Bio is just freshman year bio in college. We don't need to go into like huge detail. All right. So NAD plus is the low energy version of it that has to go get recharged. Well, where does it get recharged? Where does NAD plus get energized again to make NADH? I'll give you a clue.
Krebs cycle.
Krebs cycle. All right. Okay, so then it goes into ubiquinone, and ubiquinone is just like a pathway that leads it to the next step. There's another complex, a smaller complex we call complex 2 or succinate dehydrogenase that takes FADH2 from the Krebs cycle and strips off 2 hydrogens. Okay, so 2 hydrogens are going to pop off with this one and 2 electrons. So this only says 1, but it's supposed to be 2 electrons that then feed into ubiquinone also. And they join the process, they join this electron transport chain in ubiquinone, then going to complex 3. All right, there's no hydrogen pumping going on here, but there will be over here. All right, so you might wonder, why is this called succinate dehydrogenase? Well, if you remember from the Krebs cycle, state fair, succinate, fumarate, and right before fumarate is where FADH2 is produced. So succinate is closely involved with the production of FADH2, hence FADH2 comes in here at succinate dehydrogenase. So it all kind of makes sense with how it relates to the Krebs cycle. All right, over here the electrons pass through this thing and they power this machine, this complex 3, which is called cytochrome C reductase, in order to pump another proton, or actually, it's not just one, it's actually several. So it's not just one, technically it's more than one, but let's not worry about that right now. Protons get pumped in over here also. The electrons are then fed. And by the way, I've lost track of how many electrons, like four, is it 4 electrons? I don't really care. It's a bunch of electrons at this point. It's just a continuous stream of electrons. Let's not worry about the number. But it is 2 here and it is 2 here. But they just keep flowing. You know, they don't wait for each other to come in pairs or anything like that. Those electrons pass through cytochrome C, which is a gateway to allow the electrons to get to cytochrome C oxidase. That's unit 4 here, or complex 4. And from there, they leave the electron transport chain and they go into the matrix down here. At that point, what's going to happen is oxygen gas, the stuff you breathe in, is going to combine with those electrons. It's going to grab some hydrogen ions out of the matrix fluid that's in there, and it's going to create water. Now, this reaction isn't balanced. Don't worry about that right now. I'm going to balance it in the other drawing, and it's going to produce water. But this is the reason we need oxygen right here. That's to keep those electrons flowing. It's to accept the electrons out of the electron transport chain. It is the final electron acceptor. Super, super important. It's not aiming here. That's not what I'm aiming at. I'm aiming at this oxygen here, okay? It's the final electron acceptor. So we have to have oxygen to keep this flowing, because if there's no oxygen, the electrons get stuck right here. And everything backs up all the way back to the beginning. Everything backs up, which means this will no longer get pumped. This will no longer get pumped. This will no longer get pumped. Notice at cytochrome C oxidase, we also have hydrogen getting pumped into the intermembrane space. So all of that hydrogen pumping is going to stop, which means the whole point of the electron transport chain is going to cease to exist. It's not going to function anymore. Well, what is the point? Well, the point is this. This huge hydrogen ion concentration here wants to push back into the matrix. It wants to go this way again. And the only place it can get through is at ATP synthase. So the hydrogen ions, the protons, they come in here and they literally cause ATP synthase to spin over here. They actually go through like a little merry-go-round carousel kind of spiel and make it spin. This is a protein molecule in a living cell that spins, which is super weird because stuff usually doesn't spin in living things. We don't usually have wheels on gazelles running away from, you know, a cheetah with wheels on it. We have legs and wings and claws and things like that. Living things usually don't have wheels, or spinny things. And this is something that actually spins, which is super weird and really interesting. That spinning energy, that kinetic energy, is then transferred into these catalytic knobs down here for them to make ATP. So ADP and inorganic phosphate is going to get combined to make ATP. So I kind of lied. I said I was going to go through this super fast, and then I was going to do it slowly for you guys to draw. I kind of spent a lot of time on this, so I don't really want to do it slowly, but I'll do it one more time here. You guys are going to help me. All right, complex 1, just shout out what that's called.
NADH dehydrogenase.
Okay, and what comes in there? It's in the name. It's in the name. What comes in?
NADH.
NADH, yes. So if you could yell it at like a choir of angels, that would be the best. If I could hear all the lovely voices yelling it, that would be just so heartwarming, I would say. Okay, now the hydrogens I'm going to draw in later because I want to draw those in a different color. I'm going to draw the hydrogen ions going through. I'm going to make those blue just so it looks a little cooler, okay? So we're going to make our electrons go in here. There's my 2 electrons. And they're going to pass through and they're going to go into this complex Q, or you can call it U. It's either Q or U. Either way, U just stands for ubiquinone. Q stands for coenzyme Q. It's the same thing. There's 2 names for this thing, but ubiquinone is fine. All right, so those electrons are going through there, yay. And then down here we have what coming in? Complex 2.
FADH2.
Thank you. That's going to make FAD and 2 hydrogens. And it's going to send 2 electrons this way. So now we have a continuous flow of electrons that passes through this complex 3. What's that called again?
Cytochrome C reductase.
Cytochrome C reductase. Hey, you guys, this is cytochrome C, right? This is called cytochrome C reductase. Why does that make sense based on what you know about oxidation and reduction? What is, when something gets reduced, what are you doing to it? When you're reducing a molecule, you're giving it something or taking something?
Giving
Taking.
electrons.
Wait.
Say it louder.
Giving it electrons.
Giving it electrons. That's exactly what this complex 3 is doing. It's cytochrome C reductase. It's reductasing cytochrome C. The name totally makes sense. It's perfect. It's perfect. So it's giving electrons to cytochrome C. Cool. Not a very creative name, to be honest. When they had to come up with a name like ubiquinone, that's a creative name. That just screams, you know, class. Cytochrome C reductase, it's kind of like, oh, you just called it, you know, what it does. It's like, instead of calling me Mr. Volk, if they would have called me guy who drones on and puts people to sleep, if that was my actual name, that's kind of what this is like, basically. All right, and then we have complex 4 over here. The electrons pass from cytochrome C into complex 4, and that is called cytochrome C oxidase because it is oxidasing cytochrome C, which means it's doing what with the electrons from cytochrome C? Taking them from cytochrome C. So the name totally makes sense too, even though it's kind of a dumb, boring name. So now we have 2 electrons coming through here. I'm going to write the 2 again. So I'm back with numbers again all of a sudden. And I'm going to balance this. Have you balanced equations in chem already? Probably not, but seniors, you guys know. So I'm going to do a really weird balancing that the chem teachers must never find out about because they would yell at me and they would have a conniption. You're never supposed to do this when you do this in chemistry, but here we're going to cheat. We're going to say 1/2 oxygen, 1/2 O2, which just gives us O. But the reason I don't want to write O is because O is an oxygen radical and it's really like horribly destructive and toxic. So I don't want to put O anywhere in my cell. That's really bad. But 1/2 O2 just implies that really we're going to do this twice with 1 O2. And if I want to look at how it would work once, I'm going to use 1/2 O2. Okay, so 1/2 O2 plus 2 hydrogen ions plus 2 electrons, oh, this is all going to work out, is going to make, guess what? What did that just make?
H2O.
Oh, perfect. Okay, so we've made H2O. That's exactly right. So really, in reality, it wouldn't be a 1/2 O2. It would be 1 O2 and 4 hydrogens and 4 electrons makes 2 H2O. That's all. I just like the way it looks this way because we use 1 oxygen. Oh, I'm sorry. We use, we make 1 water and we take the 2 electrons, you know, like from NADH or whatever. So I kind of like how that works out. Okay, now I'm going to change my color. I'm going to put in the hydrogen. So we got a hydrogen here, or actually several. It's not implying it's just one. Gets pumped in. We got hydrogen ions here getting pumped in. And we have hydrogen ions here getting pumped in, which means we have a huge concentration of hydrogen here. And those are going to diffuse now back. Going to make this thing spin round and round. And that then is going to power ADP to make ATP. And I'm going to put little like a halo glow around it because why? Is ATP a big deal?
Yes.
Yes. Abigail Stroshan, you couldn't have phrased that better. That was perfectly, very eloquent, perfect answer. Anyway, ATP. That's what it's all about. That's why we do any of this stuff. This is why we breathe oxygen. This is why we eat food. This is why you go to school. This is why I come to work. This is why people, whatever people do, we do for ATP. This is it. This is what it's all about. So if you've been wondering, like, what's the meaning of life? Why am I here? Why am I getting up to go on Zoom and listen to this guy drone on forever? It's because of ATP, because you want ATP. You might say, well, what does school have to do with ATP? Well, you're going to school so you can get smart so you can get a good job, so you can make a lot of money, so you can buy food, so you can break it down, so you can make glucose, so it can be split in glycolysis into pyruvate, so the pyruvate can go into the Krebs cycle and get broken down into CO2 and make NADH, and then the NADH can pump protons into the intermembrane space, and then the intermembrane space allows the protons to diffuse back through ATP synthase to make ATP. Are you convinced yet? I don't have to say that again, do I? I don't have to repeat that, do I? My goodness. ATP is lame. That was fast for what? The was fast for what? I don't even know. Is that like teenage talk? Like, I don't understand like how you guys talk or whatever. Like simping, I don't know what that is either. Murdered my parents, it's prehistoric. Wouldn't the hydrogen be higher on the outside? Is that why it wants to go down? We're not talking about outside. We're talking about into that little space in the mitochondrion. Okay, so like this right here is the space. This is the matrix. So if you take a look at the mitochondrion once again, if you look at the inner membrane, we're not out here. That's not where we are. We're here. We're right here. You know what I mean? This is where the hydrogen ions build up, right here. Leilani, are you getting this? Okay, cool. Let's see what else. Taking bra. Is it why nobody likes Noah? I think that's because he's ugly. ATP is lame. No, that was fast for what? Yes. Okay. I think we've covered everything. If you cut off oxygen, everything stops. It comes to a grinding halt. You're not pumping hydrogen anymore, and you stop ATP synthase. A little bit of hydrogen will be in here already. That'll diffuse through, and this is going to come to a stop very quickly. ATP will no longer be produced, and your cells are going to start dying very, very, very quickly. Which cells in your body die first? If you cut off oxygen, which cells die first? Brain. Yeah, unfortunately, it's not like your toe cells or your butt cells. Unfortunately, it's your brain cells that die first because they have a very high oxygen demand all the time. The brain is always active. Even when you're asleep, the brain is doing stuff, and it always has a really high oxygen demand. A huge percentage of the amount of oxygen, and I don't know what the number is, don't ask me, but you know, it's a big percentage of the amount of food that we take in and the amount of oxygen that we take in is used up by the brain. Well, for some of us, the brain uses a lot, but some people maybe not so much, who knows? Anyway, so that's unfortunate because after a couple of minutes of not having oxygen getting supplied to these cells, they start dying. There's irreversible brain damage, and that is a big problem. Anyway, all right. Any questions on this? Sweet. No questions? Moving on. Here's just like a break, like ATP synthase is way more complicated than I make it. You know, I just draw it like an upside down mushroom. It's a lot more complicated. It's got this rotor that spins. It's got the stator on the bottom. It's got no, that's actually on top. It's got the catalytic knobs on the bottom that help with the ADP going to ATP and all of this stuff. We're not going to worry about the details on that. That's not, you don't need to know that, but you should know what the function of the ATP synthase is and how it depends on NADH to drive the protons up there and how the protons make it go. It's like a water mill. It's like the mill on the stream where the wheel turns, and that's the mill wheel turning, and the water is basically proton current. So it's a pretty good analogy. Instead of grinding grain with the mill wheel, we're making ATP. So it's not a bad analogy. All right, we're not going to watch the pretty video. You can watch that on your own because it's really slow. It's like all in slow motion. It takes forever. It's just a fancy way to diagram it out. This is the way your textbook diagrams it. It's nice. I love it, but it's just harder to reproduce. So I try to simplify it with my drawing. Although this is pretty good. Hey, they do the half oxygen too. Check it out. So I'm not the only idiot that does that. Look at this. Ha. I've been legitimized. Okay, so terminology-wise, this is where people can trip up. People can memorize this whole thing. They can understand it perfectly. They totally know what it's all about, what the purpose is. And then on the AP test, all of a sudden they're like, discuss chemiosmosis. And people are like, well, I don't know what that is. Chemiosmosis is just simply the process of hydrogen ions diffusing through ATP synthase to allow it to make ATP. That's it. So terminology actually does matter, because if they use that in a question and you don't know what chemiosmosis is because Volk never said it, then we got a problem. You might understand the whole process, but it's good to know, this happening right here is chemiosmosis. It's stupid name again. Chemi just means chemicals, and then osmosis just means like, usually it's the diffusion of water. They're not even talking about water here. They're talking about hydrogen ions. So it's not actually technically truly osmosis. So it's a dumb name, but this is the name that's accepted. That's what everybody calls it. Electron transport chain is all of this, where the electrons are, pumping the protons. That's all of that. And then all of it together, everything is oxidative phosphorylation. Oxidative because we need oxygen. And we're like, oxidizing NADH and stuff like that. We're oxidizing it because we're taking away electrons. Phosphorylation, because we're sticking a phosphate onto ADP. We're phosphorylating ADP. All right, so oxidative phosphorylation, I talked to you guys about before. It's when you take free phosphates floating around in the solution in the matrix and stick them onto ADP to make ATP. Substrate level phosphorylation is when you make ATP where the phosphate was stuck on another molecule first. It wasn't free. And that's how we make ATP in the Krebs cycle, and that's how we make ATP in glycolysis. So that's the minor way to make it. The major way to make it is this. This here is the way to go. I love how I'm pointing at my monitor and you can't even see where I'm pointing. Like, I'm pointing at crap you can't even see. Like, what am I even doing? I should be pointing at this. Okay. Do you guys notice that I do that? Like, I'm pointing over here and yeah, whatever. Sorry about that. Any questions on this here? This picture is nice. It's just a little more complicated, so it's pretty good. All right, those you guys that are hearing this stuff for the first time because you didn't have honors bio, is this overwhelming or are you doing okay? Give me some feedback here. Or you can like message me or whatever, or you can say it right now. I mean, up to you. Because there's like 3-4 people in here that didn't have honors bio, and this is like the first time you're getting this. I'm assuming you didn't do this in regular bio. I don't know. I'm pretty sure you didn't. Nobody's answering. So say nothing if you're fine and you're good and you're feeling confident. Say nothing. Perfect. I did that with my regular bio. I asked them, we did this project or whatever, this little activity or whatever. And then I asked them, if you love that activity, say nothing. And none of them said anything. So then I was like, hey, cool, they loved it. So I'm going to do that with you also. Yeah, somebody actually just messaged me. I'm not going to say who it was over, but what they said is kind of what I'm expecting. Like if you hear this for the first time, I'd be like, like what the heck is this? So, and they're being honest and telling me that. So if study it, go over it. The book is pretty good about it. You can watch videos on it on YouTube. You can ask me. I can go over it with you again. If you want to come at TSS and stuff like that, by all means. Just shoot me an email or whatever. If it's overwhelming, just let me know. Same with the Krebs cycle, same with glycolysis. So anyway. All right. I mean, it helps if you like this stuff. I happen to like this. So for me, this is fun. I know for normal humans like you guys, it's maybe a little bit less fun, but at least I'm having fun. So that's what counts, right? Okay, so there's a bunch of questions we should probably consider. Like, okay, like what will they ask us on the test about this if they're not going to make us draw the whole thing and not going to make us like, have it memorized? Well, what's the role of NADH and FADH2? Well, you should definitely know that. What's their job? You don't have to have it all memorized. You should just be able to say that they pump protons to drive ATP synthase in chemiosmosis. That's reasonable. What's the purpose of the proton gradient? Well, we just talked about it. What's the ATP yield? That's a tough question. Let's do the ATP count. How many ATPs exactly are we going to get from all of these different processes? That is not an easy question. The answer, by the way, is 38, but not really. The answer is 38 per glucose, but not really. And I'll show you why, but not really. So we'll go into that in a little bit of detail. I may have talked a little bit about that in honors bio, but I'm not sure in how much detail I went. And I don't know how much you guys remember even, but like speaking of the ATP count, how much of it came from oxidative phosphorylation? How much of it came from substrate level phosphorylation? In other words, how much did ATP synthase make? How much did glycolysis make? How much did the Krebs cycle make? Anyway, that's pretty easy to answer because you already know glycolysis gave us how much net ATP profit. That you should know just like that. So
2.
2 is correct. How much per glucose did the Krebs cycle give us? Per glucose, not per pyruvate. 1 or 2?
2.
2. So that's 4. I said the answer is 38. So how many are from oxidative?
34.
34. All right. All right. All right. Okay. How much ATP do we get per NADH? We'll take a look at that also. Well, we said it was 3, but is it exactly 3? The answer is no, it's not. It's actually not 3. It's something slightly different. It's about 3. How much ATP do we get from FADH2? Well, we said 2, but is it exactly 2? And the answer is no, not exactly. So we'll take a look at that. Let's now just say, let's just say for fun that NADH gives us 3. So we're just gonna like cheat and say NADH, even though we know it's not exactly 3, it gives us 3 ATP. And let's just say that FADH2 gives us 2 ATP, even though we know that's not exactly true, but it's close enough. We just kind of approximate. If that's true, let's figure out all the ATPs that we form from one glucose in respiration. Glycolysis, Krebs cycle, and oxidative phosphorylation. I want to know them all. Okay, so in order to do that, we have to keep track of NADH and ATP. We can't just count the ATPs from glycolysis because we know glycolysis also made 2 NADHs, so we have to be careful. So let's think about it. For one glucose, how many NADHs did I get out of glycolysis for one glucose? Just shout it.
2.
2 is correct. How many FADH2s did I get out of glycolysis? This is a trick question.
None.
The answer rhymes with Schmiro. 0 is correct, whoever typed it up. Sophia, thank you for that. So it's 0. How many ATPs did I get from glycolysis?
2.
2 only. All right, so now it gets tricky. My subtotal, that's not really a subtotal, but this is really the math. Let's do math here. I don't know why I called it subtotal, but whatever. Let's do some math. We have 2 times 3 ATPs from NADH in glycolysis that later is going to go to oxidative phosphorylation, the electron transport chain, and it's going to make us ATP at ATP synthase. So this didn't directly come from glycolysis, but originally that NADH came from glycolysis. So we're going to have 2 times 3, which is 6.
6.
Plus 2 here. So this is really 2 times 3 over here. That's 6 plus 2 is 8. Everybody agree? Okay, I hope you agree. We got 2 times 3, that's from NADH here. And then plus the 2 we already got from glycolysis makes 8. So you might say, well, but glycolysis didn't make ATP. Yeah, you're right, it only made 2. But it gave us 2 NADHs, which could feed into the electron transport chain to make 6 more. All right, cool. Then there's just a space here. Okay, Krebs cycle. How many NADHs per glucose do I get? This is tricky, be careful. Well, we got the Krebs cycle, remember? We fed in, we got one here. It was yes, no, no, yes, yes, no, 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 yes. So how many is that? 1, 2, 3, 4 per pyruvate. But I asked per glucose. There's 2 pyruvates and a glucose. Somebody shout it out, let's go. 2 times 4. Let's go. 8. Okay, so 8 NADHs per glucose from the Krebs cycle. How many FADH2s does the Krebs cycle make? Well, FADH2 only comes out once over here, but the Krebs cycle goes around twice per glucose, so we get 2. I know everybody was saying 2. I wasn't going to wait, though. All right, how many ATPs do we make per glucose in the Krebs cycle? ATP comes out down here. It's just 1, but it goes around twice per glucose, so we get, shout it. Oh, people are typing. Okay, well, I get confused with the numbers because I know I just see a ton of numbers on the chat, and I don't know which one people are answering. So if you set it into the mic, it would be better. We get 2. Now let's do the subtotal. 8 times 3 is 20. Wait, no, it's 8 times 3. 8, 16, 24. Yeah, 24. Oh my God, I was freaking out. Am I doing the math right? Yeah. 3 mal 8 ist 24. Yeah. Okay, 2 times 2 is
4.
4.
amazing. Plus the 2 here, so we got this 24, we got 4 here, we got 2 here, makes
30.
30.
30.
Add them together, 38 ATP. Everybody clear? I want you to be able to do that by yourself. If I give you a fraction of this blank, I want you to be able to fill it in. Like I want you to be able to come up with, like, if I say, for 1 pyruvate, how many NADHs does the Krebs cycle make? Oh, it's 4. For 1 glucose, how many NADHs does the Krebs cycle make? Well, it's 8. Out of those 8 NADHs, how many ATPs can I get? Oh, it's 24. Break down how many, how do we get 38 ATPs? Well, here's how that breaks down. You don't have to set it up my way with this table, but this is one trust me, I've redone this table 19 times. It was much worse in the beginning. This is the best one I came up with so far, but I'm sure it can still be improved if you want to make a better one. 10,864,643 is how many ATPs per second the cells in your little finger make. That is the question. You gave me the answer. Good job. All right, moving on. So here's just the solution. But it's actually more complicated than that. We simplified because we said 3 ATP per NADH, and we said 2 ATP per FADH, and that's not actually true. So now let's take a look at something that I have not told you yet. Let's actually take a look at what is going on here with these hydrogens. We're pumping through once, we're pumping through twice, we're pumping through thrice. So a whole lot of people think, oh, it's 3 hydrogens per NADH. And that's not actually true. It's actually 10 hydrogens get pumped through from the energy from 1 NADH. So see how that's a little bit more complicated than we thought? It's actually 10 protons getting pumped through. And FADH2 only pumps through 6 protons. FADH2 only has the energy to pump through 6. Why is FADH2 less energetic? Why does it provide less, contribution here? Look at where it jumps in. It jumps in at step 2. So how many times does it pump hydrogen? 3 times or twice? It only pumps hydrogen 2 times, which means it's going to provide less energy overall to the hydrogen contribution. Am I allowed to talk about church and religion here, or is that going to be offensive? Because this is a perfect analogy. And this was just a reading at my wife's church where I went a couple of weeks ago. This was one of the readings. Can I talk about this, or is this going to be like, am I going to get fired? Oh, you guys gave me permission, so I'm going to do it. So in the reading, it was like some kind of a parable that Jesus was telling the disciples or whatever. And he was saying that he was talking about the guy who needed work done on his farm. And he had people come in at 7 in the morning and they worked till 5 and they got paid a lot of money or whatever. But some people came in at noon. Some people came in at noon and they worked till 5. They worked half the amount of time as the other guys and they got paid the same amount of money. You know what I mean? Well, you know, in reality, the guys that came in at noon only did like half the work of the guys that came in at 7. So NADH is the guy that comes in at 7 or at noon.
7.
At 7. He does the work, or he or she, or whatever. I don't want to be sexist or whatever, but they do the work the entire day. They're pumping protons 3 times. FADH2 is the worker that came in at noon and only worked a couple hours and then got, you know, expected the same pay. Well, you know, something, they didn't produce as much stuff. So when I was sitting in church and they were talking about that, the first thing I thought was like the friggin' electron transport chain. That's going through my brain. I'm actually supposed to be thinking about like salvation and how to get to heaven and all that. And like instead I'm thinking about like FADH2. So that's probably a sin, and I'm probably gonna not get to heaven because of this, but I'm hoping my wife will get me there. I'm hoping she's doing enough to get me in. That's what I'm counting on, at least. So go for 2. I don't know what that means, folks. A sinner, yeah. Anyway, so I thought that was a pretty good analogy, but anyway, moving on. I'm wasting time now. I'm sorry. You should shut me up and not allow me to waste this much time. Okay, 10 protons per NADH. Oh, I don't have FADH here. So so the ATP yield will not be exactly 38 because we don't actually get 3 NADHs. We don't actually get 3 ATPs per NADH. So here's what happens. At ATP synthase over here, it takes not 1 hydrogen to make ATP. It takes 3 to 4, and that's what's so annoying. It's not 3 and it's not 4. Sometimes it's 3, sometimes it's 4. Well, how many is it? Is it 3.5? Well, there is no such thing as 3.5 protons. A proton is a whole, you know, it's a quantum thing that it has to be either 3 or it has to be 4. It can't be a half of 1. And sometimes you can actually get an ATP out of 3, and sometimes you can get 1 out of 4. So it's like luck of the draw. It's like flipping a coin, which is it going to be? And so that gives it more uncertainty in terms of how much ATP we're going to get. So if we do the calculation, knowing that, here's what happens. Let's actually do the calculation. If we have 10 protons produced per 1 NADH, so we're going to do factor labeling here. You guys do this in chem, factor labeling, if you do stoichiometry. Okay, we have 10 protons per 1 NADH. Let's assume let's assume worst case scenario. Let's assume that it takes 4 protons to get an ATP. So that's like the least amount of ATP we're going to get, because we're going to be using a lot of protons. So we're going to get only 1 ATP out of 4 protons. Okay, so we're going to do this calculation. The protons cancel out. We're going to get 10 over 4 ATPs per NADH. So that's going to be 2.5 ATP per NADH. Wait a minute, you said it was 3. Yeah, I told you it wasn't exactly 3. You might only get 2.5. That's actually worse. That's less. So it might be less than what you expected. Or best case scenario, if you get 10 protons per NADH, that's actually fixed. That's always the same. So that at least we can trust. How many ATPs? Well, what if you get 1 ATP per 3 protons? That's the best case scenario, where it only costs you 3 protons to buy 1 ATP. Well, that's really cheap. That would be awesome. So that's 10 divided by 3, which is actually 3.33 ATPs per NADH, because the protons cancel out and you have ATP per NADH left over. You like that factor labeling? Did that remind you of chemistry or no? Nobody cares. Nobody cares. Nobody appreciates anything. Okay. All right. So 3.3, 2.5. That doesn't actually average out to 3. That averages out to something less than 3. Okay, so that's an issue. So really, we're not going to get 38. We're probably going to get something less than that. All right. So that's for NADH. FADH2, same story. FADH2, we're only going to get 6 protons per FADH2. Well, Worst case scenario, 6 divided by 4, if it costs 4, is only 1.5. That's a lot less than 2. That's way less. That's 25% less. Holy crap. And best case scenario is 3 protons only, and it only costs 3. It's 6 divided by 3 is 2 ATPs. So at the very, very best, we'll get 2, which is what we're assuming we're going to get. We're saying we're going to get 2, when in reality, we actually get quite a bit less. We're only going to get between 1.5 and 2, more like 1.75 maybe on average, which kind of sucks. If we're expecting 38, you're not really going to get 38. You're going to get less. Know what I mean? Any questions on that? So that's a little bit complicated, but once you know that there's that the NADH gives you 10 protons, FADH gives you 6, it's going to take 3 to 4 protons per ATP. Then you can do this kind of math, you can do this kind of calculation. But that's one of the reasons why in real life situations, cells will not get 38 ATP out of 1 glucose. They're only going to get about 30, really. They're going to get quite a few less. You know, they say like, oh, ideal conditions. No, it's not just ideal conditions. Like you could have all the oxygen you wanted. You could have everything perfect. You still would get 38 because of this random chance stuff with the 4 and the 3. On average, you're going to get a little bit less. So then how would you answer the AP if there's variations? Well, they would never ask you how many ATP. If they say like ideally, theoretically, how many ATP, then the answer is 38. But in reality, it's less. And that's they would not ask you like in a multiple choice, how many is it? 38. No, what they would do instead is how many ideally, and then why is it different? You'd have to explain, oh, it's because of this 10 and, you know, because the 10 protons, blah, 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 and all of that. Even if you don't remember the numbers, like let's say you don't remember the numbers, you don't remember that it's 10 and, you know, 3 to 4 or whatever, you could just say, well, the number of protons per NADH, you know, is fixed. And then the amount of protons that it takes per ATP varies. You know, it's not the same each time for ATP synthase. So there's going to be variation there and there's going to be some random chance. If you said that, they'd probably give you full credit. So even if you didn't remember all the numbers, that'd be all right. How are we doing? Lucky you, I'm recording this. I'm going to put this on the interwebs so you can always watch this again. If you got confused, you can fast forward to this part and listen to me explain that so well again. Lucky, lucky. One of the advantages of virtual learning, because in the olden days, I never put videos on, I never recorded myself. I never put those videos up. So now the whole world benefits from having my videos on the internet. One of the benefits of COVID, I guess. Trying to look on the bright side here. All right. Okay. So that's it, Volk. Now we know why it's not 38. Yeah, that's not it. There's more. That's one reason, but there's another reason. There's more reasons. Oh my God, it's more complicated than that even. So this one is a doozy. I kind of like this one. This one's interesting. Okay, remember I told you that there's NADH made by glycolysis in the cytoplasm. That's not even in the mitochondrion. That's like way outside of the mitochondrion, far away. All right, well, that NADH has to get into the, it's got to get into the inner membrane, into the mitochondrion in order to power NADH dehydrogenase. Well, that sucks. How's it going to get in? Well, it can't get through the membrane. It's too big of a molecule. It's too polar. It can't get through the membrane. Oh, you put a protein shuttle in there. It'll carry it in. No, that's not actually what cells do. What they do is this. NADH will arrive here, and it'll bind to a protein that's called an electron shuttle. There's an electron shuttle right here in the membrane that'll take the 2 electrons from NADH, and NADH gets transformed into NAD+, and then just the electrons from NADH go through the membrane, and they eventually find themselves to the inner membrane to that NADH dehydrogenase. So that shuttle has to carry them across because NADH can't actually get through the membrane. Here, all of that is fine and good, because over here on the other side, let me actually make a better drawing of this. Hold on. I don't have a lot of space here, so I'm gonna, sorry, I gotta give myself a little more space here. I apologize. I should have had this set up ahead of time. All right, so I'll just blow it up a little bit and then you can see this a little bit better. Oops, that's not what I want. I want black. Okay. So let's say here's the wall of the mitochondrion. As a matter of fact, we're going to just say it's both membranes together. It's the outer membrane and the inner membrane. And you got this shuttle that like spans both membranes here, okay? And ADH comes in, gives its electrons to the shuttle. This is the cytoplasm. This is glycolysis. Produces NADH. That comes in, gives its electrons. The electrons go across, right? 2 electrons. Over here on the other side, there's NAD+. Okay, that's cool. NAD+ will grab the electrons and make NADH. Oh, nice. Okay, we're good to go. This can go ahead and do its 3 ATPs. Well, it's really 2.5 to 3.3. Well, let's just say it's 3. Okay, so that's nice. Everything's fine then. What's the problem? Here's the problem. Sometimes the shuttle is going to take the 2 electrons and it's going to actually give them to FAD. So there's a little bit of FAD floating around here, not as much as NAD+, but there's some FAD floating around because remember, FADH2 gives the electrons to complex 2, and it does a little bit of the pumping of the protons too. So there's some FADs which are spent, low energy, they're floating around. And sometimes just by accident, by just random chance, FAD grabs the electrons. And if FAD grabs the electrons, it's going to form FADH2. So if you end up with NADH, hey, lucky day, we get 3 ATPs. Everybody's happy. But if you end up just by random chance with FADH2, you're only going to get 2 ATPs. It's kind of like when somebody's bringing in like the mini subs from Jimmy John's. Somebody's bringing in like a big plate of mini subs and everybody goes up there and grabs a random sub and you got the Italian special and somebody else gets the ham and cheese. And then Timmy walks up and all there's left is veggie and he grabs the veggie accidentally and then just gets the veggie sub. Gross. Actually, Jimmy John's veggie subs are pretty good. I've had them. They're not that bad, even though I'm not a vegetarian. They're pretty good. They got guacamole on them. But anyway, so you end up with the crappier one. That's what's happening here. So if you just by bad luck got FAD coming in here, you're only going to get 2 ATPs from this NADH. NADH is supposed to give us 3, but it only made 2 because just by bad luck, you got FAD made over here. Bummer. Sucks to suck. Okay. Does that make sense? So now this picture over here shows that pretty nicely. This shows how NADH comes in from glycolysis. It's either going to make NADH or it's going to make FADH2. If it makes NADH, we're going to get, 3 or 6 because it's 2 of them. If it's FADH2, we're going to get less. So there's some subtraction going on from your 38 right there already. For the 38, we assumed these would all get 3 ATPs. We assumed that we're going to be 3. Well, if you're only getting 2 some of the time, you're not going to get 38. You're going to get less. So that makes sense. All right. I think that's why they wrote 36 here. It's 36 if you're getting FADH2 made, and it's 38 if you're getting NADH made. So I think that's the reason they said the 36. But this kind of breaks down the count also. 2 ATPs from glycolysis, 2 ATPs from citric acid, 32 to 34 from oxidative phosphorylation from all the NADHs and FADH2s. Add them all together, you get 38 or 36 if FADH was in play here. Give me a little feedback. See, I'm all like proud of myself here. I'm sitting here thinking, wow, Volk, you explained that so well. You're so amazing. But on the other end of the thing, where you guys are sitting, maybe it doesn't make any sense. So you guys got to let me know if that didn't make any sense. And if if you understood it or if it was fine, or kind of give me a little bit of an idea if I can move on. Remember, my goal is to finish this here today. Still have a lot of time, right? 1 o'clock. Oh, I got 12 minutes. I'm gonna have to talk fast now. Well, I don't see any questions popping up, so we're gonna keep going. There's some other reasons why, some other minor reasons why you get less than 38. So we already talked about this. This has to do with that 10 protons and 3 to 4 business. 3 to 4 protons per ATP synthase. So that has to do with that, where it's like 2.92 and 1.75 on average, not 3, not 2. So that's way less. We talked about the electron shuttle. So we're either going to get FAD or NAD plus coming in. That affects it. We just talked about that. There's actually proton leakage into the matrix. So if you're looking at the intermembrane space, you know, where you got ATP synthase over here, you know, blah, blah, blah. Some of the protons actually bypass ATP synthase and they just kind of accidentally fall back into the matrix. They kind of sneak through in places where they're not supposed to. That's minor. There's not a lot of that going on, but it's pretty well sealed. But there might be a little bit of leakage, excuse me, that where it's all supposed to go through ATP synthase, but some of them don't. Also, pyruvate has to get into mitochondria and you have to pay to get pyruvate in. So that's going to cost a little bit of ATP to get pyruvate in. So that's going to reduce your overall ATP profits. Okay, so any of these are reasons why ATP is less than 38 at the end. Realistically, it's about 30 to 32 in ideal situations. All right. So that slide kind of gives you a summary of all the reasons. All right, I'm going to go pretty quickly through this because this is not, I think this is easy to understand. I hope it is. We're going to talk about anaerobic respiration. So like, what if there's no oxygen? In the first situation, we're not going to talk about humans or animals. We're going to talk about lower complexity creatures. What am I trying to say? Low level primitive creatures. Yeah, we're talking about primitive creatures like single-celled creatures like yeast and things like that, amoebas and things like that, bacteria and stuff like that. They can actually live, some of them, I should say, can live without oxygen. Some of them have mechanisms that can live without oxygen and survive for a while in like a dormant state, like an emergency kind of a state where they don't do a lot, they don't move, they don't reproduce, they just kind of try to survive the low oxygen states. There's some that actually thrive under low oxygen states. Some bacteria are anaerobic. That's not what we're talking about. We're talking about like normal, like a cell like yeast, for example. If there's no oxygen, what yeast will do is it'll still keep glycolysis going, but everything else has stopped. See, the Krebs cycle will stop, the electron transport chain will stop, all of that will be stopped. So it'll try to keep glycolysis going and just make a little bit of ATP just to survive. Here's how it does it. Here's what it does. It keeps glycolysis going and gets that little bit of ATP, just 2 per glucose, by taking the pyruvate and actually breaking it down and wasting it. The pyruvate is going to get converted into acetaldehyde, and acetaldehyde is going to get converted into ethanol. So ethanol is going to be a waste product. Well, ethanol still has a bunch of, cutting out. I'm sorry. Okay, I didn't know that was happening. So was that happening the whole hour? Probably, right? My video, okay, it just started. The video that I'm recording, yeah, it's telling me slow connection. So it actually is just starting for me too here. My video should be perfect, I hope, because I'm recording it on the computer, not on the cloud. So if for the last 8 minutes, this cuts out and you're not getting all of this, please watch the video, okay? So I'm gonna keep going and I'm gonna trust my video will be in good shape, hopefully. All right, so the ethanol is gonna get wasted. That's a waste product. And what's even worse is all this NADH is wasted now. As a matter of fact, we have to get rid of the NADH because if the NADH builds up and there's a shortage of NAD+, this will stop. If there's no NAD+ to feed into glycolysis, this will actually stop and you won't even be able to get this to ATP. So you have to get rid of the extra NADH, sadly. You can't save it. You have to get rid of it. And that's what happens. The NADH drives this acetaldehyde reaction to ethanol. And it supplies NAD+ to glycolysis to keep it going. So the yeast cells can only do a little bit, they only can get a little bit of ATP through this, and they can only do this for so long, because if they're in a closed environment, the ethanol builds up and this will kill them. The ethanol will actually kill them because it's toxic. Just like ethanol will kill you if you drink too much ethanol, it's a poison essentially, and it's going to kill the yeast. The other thing that happens is CO2 is produced, and this is what makes alcoholic beverages like beer and sparkling wine sparkly. It makes it bubbly. I'm not trying to tell you to go out and drink just because I'm talking about beer and wine. I'm just telling you that's why champagne and beer and things like that have bubbles, because of the CO2 that's produced in this process from pyruvate to acetaldehyde. This is 2 carbons. This is 3 carbons. The extra carbon leaves as CO2. All right, so that's ethanol fermentation. The other kind of anaerobic metabolism I'd like to talk about is lactic acid fermentation. And animals and humans actually do this. Now, we're not doing this instead of aerobic because we have to do aerobic to get all the ATPs we need just to survive. Our brains and our bodies cannot function on just the 2 ATPs from glycolysis. So we're doing this in addition to running aerobic Krebs cycle and oxidative phosphorylation at 100% when we're running really fast or we're working out really, really hard at maximum capacity. We can just sprint a little bit faster if we engage in anaerobic respiration, which is this lactic acid fermentation, and we can get an additional 2 ATPs on top of the other ATP that we're already making. That tiny little extra ATP boosts us a little bit, but only temporarily. We can only do that at above 100% for very short amounts of time. We're talking about seconds, not minutes. It's rare that somebody can do this for a really long time. They'd have to be like a super elite athlete or whatever. Okay, because if you do this too long, what happens is lactic acid builds up, and the lactic acid causes fatigue and muscle pain, and it's going to shut you down. It's going to cause you to collapse if you do it too long. So then you have to slow down again or stop so that lactic acid can get broken down. It can get converted back to pyruvate actually is what happens. And that's a way to boost your performance. At the end of a race, you can do a sprint and you can run really hard and beat the opponent from Greendale or wherever, or the West Allis people. You can beat them by doing this lactic acid thing, and then you have to rest or slow down. You don't want to do this in the beginning of a long race, like a marathon or something like that, because if lactic acid builds up early on, you're going to be in big trouble. You're not going to be able to finish. Anyway, so the way that works is glycolysis, normal glycolysis, get our 2 ATPs, pyruvate, but now the pyruvate is something that builds up. We have to get rid of it. So the pyruvate gets broken down or changed into lactic acid, and in the process, NADHs get removed. So this is a complete waste of NADH. All of that ATP you could have had if it was aerobic, you're wasting. But you're getting that boost in order to win the race or to survive in a escape situation. In the olden days, if you're running away from a tiger or you have to fight for your life, this is the thing that's going to allow that to happen. The cost is that lactic acid builds up, which you have to get rid of because it causes fatigue. And the other cost is that on all that NADH and pyruvate is wasted. You don't get a lot of ATP out of it because it's anaerobic. Everybody clear on that? Hey, I still got time. We could wrap this up. Look at this. Look at you, Volk. Yeah, I'm praising myself again. Look at that. Okay, this kind of explains all that, what I just said. Then we can talk a little bit. Let me just see how much I have left. I have just this other molecules and regulation, and I think that's it. Yeah. Okay, we can do this. What about food molecules that aren't sugar? Like, what about things that aren't glucose? Like when we eat food, we eat protein, amino acids, we eat fat, fatty acids, glycerol, stuff like that. What about all of those? How do they feed into this process? Because all we kept talking about, yes, last time and today, is glucose all the time. That's kind of unfair because especially if you're like on the keto diet, you don't even eat glucose much. There's hardly any glucose in your diet and you can still survive. So how does that work? Well, all those other molecules feed into the system, and I want to show you where they feed in. So this is a blank slide that doesn't explain it to you. You might want to look at the blank slide to practice when you study this stuff. Try to figure out like what should be happening, what should be feeding in at what place. So let's look at the solution because we don't have a lot of time left. Let's take a look at that. The middle pathway is just glucose. It's just sugar, like what we're used to. Okay, so this is glucose from the diet directly, just does the glycolysis thing, Krebs cycle, electron transport, just like what we talked about. All right, but what about protein? Well, proteins get chopped up by your digestive tract into amino acids, and they get absorbed into the bloodstream as amino acids, individual little amino acids. And the amino acids have to get broken down because they have different sizes. The smallest amino acid, glycine, has only 2 carbons because it has H for the R group, and it has an amine. So the amine gets chopped off, and this is the reason why we have to produce urine. So we produce urea to get rid of nitrogen waste. So that we eliminate that as urea in our urine. That's the reason we have to do that, because this is very, very toxic. Ammonia, which is NH3, is super toxic, and we have to get rid of it. So our kidneys take care of that. All right. Then there's 2 carbons left, or there's a larger molecule left because you had a bigger R group. Well, those feed in different places. They could feed in over here as a 3-carbon pyruvate. They get chopped up into 3 carbons, or they can feed in as acetyl-CoA, which is 2 carbons. So that would be like glycine feeding in. Just 2 carbon molecules feed in as acetyl-CoA. That's 2 carbons. That makes sense. Or they feed in different places in the citric acid cycle, which have different carbon numbers. So if you have a larger molecule from an amino acid, like a 4-carbon one, it might feed in at, I don't know, succinate or something like that, which is 4 carbons. All right, and we don't need to know the details for that. That's not that important. But we do need to know that proteins can be fed into this system and can be metabolized and broken down into CO2 and, make, no, it's only 1 o'clock. I still get until 1:01, don't I? Can't you? You can give me a friggin' minute, can't you? Anna, no. I'm gonna keep talking. I'm gonna keep talking. Hey, if you leave, I'll just consider it a personal insult, and I'll be hurt and cry on the inside. Is this really what you want? Is this really? No, you're gonna sit after class is over knowing you made me cry? Is this really your goal? So let me finish. Let me finish. I'll do the regulation next time. 2 more minutes. Couple of 2-3 minutes. Okay, I'll talk really fast. Fats feed into this. Fats are going to be triglycerides, which are fatty acids, and glycerol. Well, guess what? Glycerol is 3 carbons. It feeds in at glyceraldehyde 3-phosphate. Why? Because that's a 3-carbon molecule. It even sounds like glycerol. That feeds in there. Okay, that's cool. Feeds right into glycolysis. No problem. Then we can keep going and do all the other stuff. Fatty acids get broken down by beta oxidation into 2-carbon fragments. So we get these little 2-carbon fragments, feed into acetyl-CoA because it's 2 carbons. That makes sense. Acetate is 2 carbons. That feeds into the process this way then. Okay, so fats get fed in also, and they can make tons and tons of ATP, just like glucose can. That's kind of nice. All right, and that's it. So we got carbohydrate. Oh, no. One more thing. This arrow is the only thing that's really confusing. Why would everything just go down the middle? Why are there some carbohydrates that feed in down below glucose? And the answer is it's glucose 6-phosphate from glycogen. Remember when we talked about Sutherland? We talked about glycogen getting chopped up by glycogen phosphorylase into glucose 6-phosphates. The glucose 6-phosphate is a part of glycolysis. You can feed right into glucose 6-phosphate at that step, the second step, from glycogen. So that's convenient. You can skip that first step because you have G6P already. You don't even need to start with glucose. So that's kind of awesome. That's what this arrow is right here. All right. Okay, if anybody has questions or whatever, come to TSS. I'm going to close down this because I have to open up the other Zoom. And if not, have a good week. Study the stuff I asked you to study. Get that stuff done that I asked you to get done. And we will do photosynthesis on Friday. We'll have a test Friday after this Friday. Bye. That was only 3 minutes. That wasn't so bad. People cry a lot.